So bacteria and archaea represent two different domains, but these are the two domains of prokaryotes, so we're going to look at these together at the same time. Prokaryotes, if you remember back to Bio 1, are typically small, they're always single cellular, they do not have any membrane-bound organelles, they have small circular chromosomes that are not enclosed in any kind of nucleus. And there's three typical shapes to a prokaryotic cell. The first one is spherical, which is also called cosy. And you're going to see each cell is in itself a little sphere. And they can be arranged differently. Sometimes some species have singular cells, some species have these big kind of grape-like clusters. Rod-shaped is bacilli. So these are kind of longer rods. Each cell is this kind of elongated shape. And then spirilla, spirals. And these ones show this kind of like almost wavy hair looking shape that spirals around a central axis. One of the key characteristics that we use to classify bacterial cells is the type of cell wall. So most bacteria have cell walls, which is going to make them distinct from eukaryotes. These cell walls are very different than the cell walls you would find in like plants. And there's two different types of cell walls, gram-positive and gram-negative. And so gram staining is a process developed by a scientist named Hans Christian Grams in the 19th century, and it uses different stains to stain the bacteria different colors depending on what type of cell wall they have. So when you do a gram stain, the first step you typically do is you use your gram stain, which is purple, and then you would add in some iodine, which is called a mordant. The iodine will blend in with the purple, so what happens if we have inside the cell? So that purple and the iodine would get inside the cell and stain it purple. After that, in the grain staining process, you use an alcohol to wipe away as much as you can of this purple stain. And it's at this alcohol step that we see the difference between gram-positive and gram-negative. So a gram-positive bacteria here's the plasma membrane. It has a very thick single layer of peptidoglycan cell wall. So the purple and the iodine get in here, and when the gram purple and the iodine get in here, they create a larger molecule, a violet iodine complex, and the alcohol is not able to wipe it away. So the alcohol does not disrupt this outer cell wall, and it's not able to get rid of this crystal violet stain, and so a gram-positive cell is going to end up looking purple after gram staining. A gram-negative cell, on the other hand, the first two steps are the same. We put in our gram stain, it kind of bonds with an iodine molecule, makes this big iodine crystal violet complex. But the alcohol step comes in here. Remember, alcohol is hydrophobic. And the structure of a gram-negative cell has a very thin peptidoglycan layer and then this lipopolysaccharide layer. So lipids, right? These are fats. So the alcohol comes in and it disrupts this lipid layer, and all that's left is this very thin layer. So during this third step, the crystal violet and the purple iodine stain gets removed. So we'll erase that. And then the fourth step of the gram-violet stain is a counter stain, which is called safranin, and it turns out pink. And so during the fourth step, what will happen is those pink stain molecules will come in here, and a gram-negative cell is actually going to stain pink during gram staining. Some more structures. Some bacteria will have a capsule. These are not going to be found in every single bacterial cell, but some bacteria, especially ones that are pathogenic to animals, tend to have kind of an organized capsule layer on the outside. So here's the capsule, is this thick part right here. This part right here is the outside cell wall, and then this part out here is the capsule. Sometimes the capsule is really dense like this, and so it's called a true capsule. If the capsule isn't really dense or well organized, it's typically just called a slime layer. But either way, a capsule or a slime layer is going to help this bacteria kind of stick to a surface. So this is a tonsil cell. So that would be something inside of your throat, right, your tonsils. And then this bacterial cell might be a respiratory pathogen that's going to come in, stick to your mucus lining here, and start to cause a respiratory disease. Another example of structures in bacteria are called endospores. So if you think of the word endo, that means inside. And then a spore is just kind of a small, kind of dense subject. So here's the actual bacterial cell on the outside here. And then in here, we've got this dense endospore. So what this bacteria did is it made a copy of its DNA, its chromosome, and it's enclosed it in this really tough, dense endospore surrounded by this coat. And this happens in really harsh conditions. So, for example, areas where this bacteria cell maybe isn't getting enough water. And then what happens is it develops this endospore. It actually lyses this cell. So the outside cell will actually die. But what will happen is a month later or a year later or even years later, when this endospore finally finds its way into a habitable environment, it will swell up with water and then start to become a viable cell again. So some bacteria, even though you wash your hands, use antibiotics, they can survive in harsh environments for a really long time. In order to kill an endospore in a lab, you need a high-pressure autoclave. So an autoclave disinfects bacterial products by killing them with high heat and high pressure. So it takes about 121 degrees Celsius under high pressure to kill something that has an endospore. Some bacterial cells have appendages. So these fimbriae are long appendages. These are typically found in cells that either live in colonies, so these fimbriae help them stick together, or it helps them stick to a substrate if they're trying to stay on a substrate. Sometimes they have specialized appendages called pili. Pili is plural. It's also, can see pilus. Pilus is singular. And these are typically longer than a fimbria, so a pili might be really long. And these are going to be how DNA is exchanged. So that would be how sexual genetic variation is introduced between two bacteria by exchanging DNA along a long pilus. Some bacteria can move. So about half of prokaryotic bacteria have the ability to move, and the ability to move would be an advantage as far as natural selection goes, right? If I'm looking for food, I could move toward a stimulus that means food, or I could move away from a stimulus that means maybe predator or danger or an environment I don't want to be in. Like, I could move toward water and away from being dry. And so they have these flagella that have these long spiral shapes. These are very different than the flagella found in animal cells. And there's genetic evidence that the flagella found in archaea and bacteria and eukaryota, the three different domains, actually all arose independently. So even though we see flagella in all of these different domains, we think that's just because the flagella was a great adaptation that they all came up with separately. So this would be an analogy and not a homology. So flagella across these three domains are not a homologous structure. We can use the different structures of flagella and the different proteins that they're made of to kind of see how there are differences between the three domains of life. And then some prokaryotes have more advanced enfolded plasma membranes. And because they don't have internalized like a nucleus or any kind of membrane-bound organelles, they actually use their outside membrane, their plasma membrane. So this outside membrane would fold in back on itself several times. And that's going to create the surface area needed to do things like cellular respiration or photosynthesis without needing a mitochondria or a chloroplast. They do not have a nucleus, right? So chromosomes, we typically have one big circular chromosome, and this one's folded back on itself a lot here. But it's one circular chromosome, and it's just in a nucleoid region, so it's not actually inside of a nucleus. And then something that's really common for bacteria is to have little extra pieces of DNA called plasmids. And you can see these over here. They're really, really small sections of circular DNA. Here's another one. And they aren't part of the main central chromosome, but they can do really great things for the bacteria. They can help with preventing, they can confer resistance to antibiotics so that that bacteria can survive in a harsh environment. Plasmids are also how bacteria have the ability to reproduce. Some plasmids can be transferred along those sex pilus to allow for genetic diversity to cross from one individual to another.